The cell cycle is the series of changes that a cell undergoes from the time the cell is formed, from cell division until it divides or dies. Looking at the figure on the right, we see a cell being quote unquote born as a result of cell division. That cell then grows, carries out its normal metabolism and various functions, and then at some point receives a cue that it is time to prepare for cell division. The cell then continues to grow, it replicates its DNA, makes final preparation for division, and then it starts the process of cell division, resulting finally in the dividing of cytoplasm, and then that cell's cycle ends in the production of two new cells. Of course, somewhere along the way, that cell could also have died, and that would have ended the cell cycle. The cell cycle is divided into two major periods. The first, interphase, shown in green in the figure on the right, by far represents the majority of a cell's life cycle. During interphase, the cell is growing and carrying out its usual activities and functions. The mitotic phase, shown in yellow, is the time during which the cell is actively engaged in dividing. Let's look more closely at these two periods, starting with interphase. Interphase is the portion of the cell cycle where the cell is not dividing. Often it is referred to as the resting phase. Certainly when I was in school, I was taught interphase is the resting phase. However, this is really a misnomer. The only thing the cell is resting from is cell division itself. The cell is growing, the cell is carrying out normal metabolism, so it's not really resting. Interphase itself is divided into three subphases, G1, followed by S, and then finally G2. The G1 subphase or GAP1 subphase directly follows the formation of a cell by mitosis. During this phase or subphase, the cell is very active metabolically. It's growing very quickly. This phase can last from minutes to hours to years, depending on the cell. During this time, the cell is not really preparing for cell division. In fact, cells that will never divide are said to be stuck in this phase. And if that is the case, they're actually said to be in the G0 phase. However, if a cell is going to go on to divide and get past this restriction point toward the end of this phase, the centrioles replicate. The centrioles shown here are organelles that later on you will see play a very critical role in mitosis and therefore cell division. Our cell in the G1 phase will continue to grow and will continue to be metabolically active, and we'll get to this restriction point here where it will go no further in the cycle unless it is acted upon by a chemical messenger. If the cell receives a chemical signal, typically that chemical signal is in the form of a hormone, although it could be other chemical messengers as well, it will receive the message that it is time to prepare for cell division. And it will move from the G1 subphase to the S or synthetic subphase. This subphase is all about the synthesis of new genetic material. If the cell is to divide and give rise to two new cells that are identical to the original cell, all of the genetic material must be copied or duplicated. That involves the replication of new DNA. Replication of DNA basically refers to the duplication of every chromosome. New histone proteins must also be produced because the new DNA will be wrapped around those new histone proteins in the assembly of new chromatin. After successful synthesis of new genetic material, the cell leaves the synthetic phase and moves into the gap-2 subphase. Here the cell is going to make final preparations for cell division. Enzymes that will be involved in cell division are synthesized and directed to the appropriate parts of the cell. Centriole replication is completed, as well as the replication of other organelles such as the mitochondria. And lastly, there's a check to confirm that the DNA that has been replicated is undamaged. The cell is now ready to divide and move from interphase to the M or mitotic phase.